a lesson that we are going to talk about the cell. So as usual, good morning.
Good morning, Madam Kimanzi, and how are you?
I'm very fine. So today we are going to discuss the cell. And as we are going to discuss the cell, I need you to participate so that you can be able to understand the topic very well. First question, I want to ask the question because I want us to interact. What is the cell? What is a cell? What is a cell? Yes?
A cell is the basic unit of structure and function in an organism.
She has said that the cell is a what? A basic unit in structure and
Functions in an organism.
in an organism. So that is what he said. When we talk about the cell, we are simply talking about the water. That basic unit, a functional basic unit in structure and also the function. Without that cell, an organism cannot do what? It cannot function. So when you talk about something to be basic, it is simply a water. It is a must that it should be there for that organism to function very, very well. So when we are defining a cell, you have to talk about it is a basic unit in structure and function in an organism. Are we getting the point?
Yes.
Any question? Any question on that? Have we understood what is a cell?
Yes.
Okay. So the other thing that now we have to understand, it is now we have different organisms. We have some of the organisms that have many cells, others have only one cell. So which are these organisms that have many cells, and which are these other organisms that have just one cell? We gave them the terms. What do we call them? What do we call them? Yes? They are which organisms now? Multicellular.
Yes, they have many cells.
Clever. So we are saying that unicellular organisms, they are the organisms with only one cell. And the multicellular organisms, they are organisms that have very many cells. So when it comes to these unicellular organisms, do we have examples?
Yes.
Do we know examples of unicellular organisms?
Yes.
An organism with one cell.
Yes.
Janet? We have the amoeba. Yes. Another one? Yes. We have bacteria. Any other that we know? What about in terms of kingdoms? We talked about kingdoms.
Yes.
We will be asked which kingdom comprises of organisms that are unicellular. Which kingdoms do we have? Yes? We have kingdom Monera. Another kingdom that has unicellular organisms. Yes? Kingdom Protopista. Any other kingdom that has unicellular organisms? No, we only have two kingdoms. Are we getting that?
Yes.
We have the Kingdom Monera and Kingdom Protopista. Now, what about the multicellular organisms? We have seen the multicellular organisms, they are organisms made up of many cells. Now, which are the kingdoms or the members of those kingdoms that have the multicellular organisms? Yes, Chanel?
Kingdom Plantae.
We have the Kingdom Plantae. Another one? Yes, sir. Have you?
Kingdom Animalia.
Kingdom Animalia. Do we have another one? Do we have another kingdom? Yes.
Kingdom Fungi.
The Kingdom Fungi. So when you talk about multicellular organisms, these are the organisms that are made up of many cells. So when you talk about Kingdom Plantae, we have which members? Remember, we have given examples in terms of kingdoms, but not specifically which are the members. Like in Kingdom Plantae, which are the members there? Yes, sir?
All plants.
All the plants. We have all the plants. Another one? In fungi we have? In fungi, which are the members there? In the Kingdom Fungi? Kingdom Fungi, who are the members there? Yes? We have the yeast. We have? Yes? We have the mushrooms. Another one?
Moss.
We have the moss. Is the moss under the, is it under the fungi? The moss, is it under the fungi? The moss, is it under the fungi? Are you sure? Feel free to discuss. Is the moss under that? We said we have the yeast, we have the mushrooms, another one? Yes? The mold, not the moss, it is the mold, sour. Yes, so we have the bread mold. Are we getting the point? Yes. Now we are through with now the cellular organisms and the monocellular organisms. Do you have that in our notes? Yes. Yes, we're confirming that if we have it in, we have the notes under our notes where we wrote. Then the next part now, after we are through with the multicellular and the unicellular, I want us now to talk about, because we say the cell it is a basic unit. It is microscopic. And it cannot be seen by our naked eyes. So we use a powerful instrument so that now we can enlarge the water. We can enlarge this cell. Which instrument do we use so that we can observe or we can see these cells? Which instrument do we use? Now everybody knows. Yes, let's give the name.
Microscope.
The microscope. So we will be using the microscope and we have to understand it. We have to understand the structure of a microscope, the parts that make up a microscope, and the function of each water, which part that is there. But before now we get into the parts, we have now to talk about the types of microscope. And we said we have two types of water, microscope. Which include, which are the two types of microscope. Where is Mary? Where is your blazer, Mary? You decided to be different. It is okay.
So there's light microscope.
We have light microscope.
And the electron microscope.
And the electron microscope. Let's clap already. So we have the light microscope and the electron microscope. Are we getting the point?
Yes.
And now we want to ask, there is a question behind that about when do we call it a light microscope? And when do we call the other microscope a what? An electron microscope. So what is the difference? Why do we call it a light microscope? It is like now we are discussing the differences between a what? A light microscope and the electron microscope. Do you know them or you want to read them? Do you know them?
Yes.
Now we are going to start with this row. This row you give us one, another one and another one. So it's going to represent us in this row. Yes, sir? Yes, the light microscope, it uses the water, the light to illuminate a specimen. While now the electron microscope, it uses beam of water, electrons. That is the major reason why we call it a light microscope and we call the other one a water, an electron microscope. Because the one that is using light to illuminate the specimen, it is the one that we are calling a water, a light microscope. And the one that uses the beam of electrons, we call it a water, electron microscope. Another difference? Another difference who was not answered? At the back there?
A light microscope has low magnification power, and electron microscope has high magnification power.
Yes, the light microscope it has low magnification power, while electron it has a high magnification power. You find that the number of times a light microscope can magnify or it can enlarge an object, the electron microscope it does that one more than the light microscope. So you find that electron microscope it has a high magnification power simply because it uses water, beam of electrons, or it is using electricity. So it is able to magnify more than the water, the light microscope. Another difference, let's go now. Yes, at the back there.
A light microscope has low resolving power, while an electron microscope has a high resolving power.
Yes, we are talking about now the resolving power. We'll come to define what is a resolving power. Remember, we are saying now electron microscope, it has high resolving power. And the light microscope, it has a what? Low resolving power. Before we continue, can we get to know know what is the resolving power. For our online students who will be listening to us, can you tell them what is the resolving power? What is resolving power? Yes?
Resolving power is the ability to distinguish between two structures that are very close to each other.
Yes, that ability of a microscope to distinguish two entities that are very close. It is what we call what? Resolving power. And the ability of a microscope to distinguish two close entities, as separate entities. And we think this resolving power, it is also known as, we also gave it a name. We said resolving power, it is also known as, yes, Karen?
Resolution.
Resolution. So when in exam we get resolution, we just talk about, it is the same thing as a water, resolving power. Are we getting that?
Yes.
So in which row now? This one, yes, answer, another difference.
I like to
Mm-hmm.
glass lens for magnification.
The sound, be audible.
A light microscope
Mm-hmm.
uses a glass lens for magnification,
Mhm.
while an electron microscope uses the electromagnetic lens for magnification.
Yes, very good. We are saying that the light microscope, it uses the glass lenses for magnification, while the electron microscope, it uses the water magnetic lenses for magnification. Another difference, we still have another difference, yes?
In a light microscope,
Mhm.
the specimen can be alive or dead,
Mm-hmm.
while in an electron microscope,
Mm-hmm.
the specimen is dead.
Yes, light microscope, it can view both the dense and the live, but now electron microscope, it can only view the water, the dense specimen. Why? Why? Why does it only use only the dense specimens? There is a reason to that. Yes?
It uses the specimen because they are placed in a vacuum.
Yes, because within an electron microscope, the specimens are usually placed within a water, a vacuum. And within a vacuum, we know there is no water, there is no oxygen. Another difference? We have another difference. Yes, there is another one. Yes?
A light microscope uses normal dyes, while an electron microscope uses complex dyes.
Yes, the light microscope it uses normal dyes. We know that we will talk about these dyes and we gave example of our water, iodine solution. But now when it comes to the electron microscope, it uses the water, complex dyes in order to do water. So that now we can make the observation distinct and clear. Do we have another difference? Any other difference that we have between light microscope and electron microscope? Who has not answered? Yes, Chanel?
Under the light microscope, the specimen is placed on a slide, while in the electron microscope, the specimen is placed on a vacuum.
Yes, now under that difference, we usually talk about mounting. We usually mount the specimen on a water, a slide that is usually placed on a stage. But now when it comes to the electron microscope, the specimen it will be mounted on a water, on a grid that is usually placed within a water, a vacuum. That difference for you to get it right, you have to be specific. We are placing now a specimen on a slide, and that slide it will be placed on a water stage. Are we getting the point? Yes. But within electron microscope, now we are talking about the specimen it will be placed on a water, on a grid that will be placed within a water, a vacuum. Any question? Any difference that we have not understood? So to our online students, if there is any difference that you've not understood, please comment there, ask the question, the teacher will get back to you. So that was our end of our lesson. How was the lesson? It was nice. Yes. So this is my 1 Peak 2023, and you can see they are very lovely and they are very beautiful. We will be engaging you more into our episodes. So each class will be posting to you so that you can follow us. Please to do what? Like,
share,
follow, and
subscribe.
Bye bye. Say bye.
Well, our biology lesson has been well. We have enjoyed our lesson and we have learned many things. And I wish you a Merry Christmas.
Merry Christmas to you. People are not saying very
Merry